Hello and welcome. Today we're going to be filling in some notes and doing some practice about one-factor and two-factor crosses. One-factor crosses, of course, are where you're looking at one gene at a time. Two-factor crosses are where you look at two genes at a time. You'll also hear those called monohybrid and dihybrid crosses, meaning one gene and two genes at a time. Let's begin with one-factor crosses, problems that involve only one gene at a time, like flower color or seed shape or plant height, but only one gene at a time, only one trait. All right, the example that we're given in part A says, in peas, green seeds are dominant to yellow seeds. All right, green seeds are dominant to yellow seeds. Two heterozygous green-seeded plants are crossed or mated. And then it has some blanks there for us to fill in. The first one we need to fill in is the gene and the gene traits. And that's actually written over to the side here. It says gene and has a blank. Now let's see. It says that green seeds are dominant to yellow seeds. And so what we're talking about is seed color. And we're talking about green seeds being dominant to yellow seeds. Now we'll need to represent, that's an N, there we go. We need to represent each of those with a letter, and the dominant gene needs to be represented with a capital letter. Since the green starts with a G, we'll represent it with a capital G. Now we need to use the same letter for the recessive gene so that we recognize they are the same trait. So even though yellow starts with a Y, we're going to represent yellow with a lowercase g. So capital G for the dominant trait green, lowercase g for the recessive trait yellow, and that is how we fill in the gene information. Then it has P and it has two blanks. This is to fill in the parental cross. So we're going to fill in the parental genotypes. It says two heterozygous genes. Since the problem says to cross two heterozygous green-seeded plants, remember that heterozygous means that the heterozygous means that the two gene letters are going to be different. In other words, one will be capital and one will be lowercase. So a heterozygous genotype is capital G, lowercase g. And since it says to cross two heterozygous plants, we would be crossing one capital, one lowercase, one capital, one lowercase. And that would be our parental genotype. At that point, then we will fill in our monohybrid cross square. And so I'm just going to sketch the square real quickly. Ooh, that one line over there is not very good. Just ignore that. All right. Now, this parent could donate a capital G gamete or a lowercase g gamete. So I will write those along the side of the square as possible gamete choices. This parent could give a capital G or a lowercase g. Again, those are the possible gamete choices, so I'll write them there. Take a second and fill in your chart. We see capital G, homozygous dominant. Capital G, lowercase g, heterozygous. And little g, homozygous recessive. The question says, what are the chances of getting a yellow-seeded offspring? Well, when I look, this is green, this is green, this is green because of their dominant traits. Well, this one is yellow because it is homozygous recessive. And so that is 1 out of 1, 2, 3, 4. So 1 out of 4 chance, or 25% chance of getting a yellow seeded offspring from two heterozygous parents. All right, that is how we work one-factor crosses. Hopefully that was a helpful review. And now let's go ahead and move into part B, two-factor crosses. These will involve two genes at the same time. So maybe this time seed color and also plant height, or seed color and also seed texture, or plant height and and fruit shape. It could, who knows? But any two traits. So we're looking at dihybrid, which will be two traits. First, a quick review. As we look at B1, it says to identify whether something is a genotype or a phenotype. Remember that if it has letters, which represent the genes, those are a genotype. And if it has a physical description, such as tall or yellow, those are phenotypes. Again, physical description, phenotype, they both have that f kind of sound. So phenotype, physical description, words, tall or yellow. Genotype, gene letters that tell you what information is on the chromosome. So that's genotype and phenotype. Go ahead and pause the video and fill in part B, number 1. Now for part B, number 2, we're going to indicate whether something is heterozygous or homozygous. Okay, if it is heterozygous, remember that the word hetero means different. And so if you have, say, a capital D and a lowercase d, those are different from each other. And so those are going to be heterozygous. Well, what if you had something that was the same? Okay, the prefix homo means same. Okay, and so if it were two capitals or two lowercases, those would be homozygous. Okay, so this is heterozygous. And both of these are homozygous. Now, since both of these are homozygous, we need one more word. Both are they are the same, but they are also both capital. And so I would say homozygous dominant. And since these are the same but both lowercase, I would say homozygous recessive. Ooh, remembering how to spell recessive, it's a hard thing to do. Okay, so homozygous dominant and homozygous recessive. So take a moment and fill in on part B, number 2, whether those things are homozygous, heterozygous, and homozygous dominant or recessive. All right, as we move on to part 3, we're going to write the genotype for the following phenotypes. So when we're given a phenotype or a physical description, we're going to write the genotype or the gene letters. The first one, part B3A, says a short plant with white flowers. Now, when I look at the trait, at the table of traits just to the right, I see that plant height is represented by T's for tall and short. Now, since short is the recessive trait, in order to have a recessive phenotype, I will have to have a homozygous recessive genotype. Because if there were a capital T at all, that would be what was noticed and observed. For white flowers, again looking, and I see flower color is purple or white, and so we'll be using P's to represent that. But again, homozygous recessive, because if we had a capital P, then the flower would be purple. So in order to have a white flower, we will have to have a homozygous recessive genotype. Now we're looking at both of those traits at the same time, and so we need to write them, and I'm going to write them alphabetically. So P's come before T's, and I'm going to write P, P, T, T. And that will be the genotype for letter A, the short plant with white flowers. Now, what about, let's see, so I'm just going to label that A. What about B? Heterozygous for both seed shape and seed color. Okay, so seed shape and seed color. And it says heterozygous for both. Now in this case, let's see, seed shape is represented by round or wrinkled, and it's represented by R's. Remember that heterozygous means we have one uppercase and one lowercase. Seed color, of course, is green and yellow, represented by G's. Again, one upper and one lower, because we were told that they were both heterozygous. When I put those together, again, I'm going to write them alphabetically. I'll have my heterozygous for seed color and also heterozygous for seed shape. And that will be that. Part C. Let's look at part C. A homozygous tall plant with wrinkled seeds. Homozygous tall. So homozygous tall. Okay, and since tall is the dominant trait, that will be capital T, capital T. Homozygous, same. Tall, capital T. So capital T, capital T. And wrinkled seeds. Remember that wrinkled is a recessive trait and is represented with an R, so it will have to be homozygous recessive, little r, little r. Again, let's put those together. And I'm going to, again, write them alphabetically and have R, R, T, T. Take a moment and see if you can fill in D through G. All right, so let's take a look at part B, number 4. For each problem below, do steps 1 through 3, that is, write the gene, the dominant and recessive alleles, and the parent cross. Okay, so let's look at part A. In mice, the ability to run normally is a dominant trait. So I'm going to write normal running. So that is the dominant trait, and so I'll represent that with an N. And then it says that the recessive trait causes mice to run in circles, and they are called waltzing mice. Okay. I see that that your page actually doesn't use an N; it uses an R. So I'm going to change and match what's listed on your page. Normal running, and the recessive trait is waltzing, which will be a lowercase r. A waltz is a dance where the dancers move around in circles, and so we have the normal running mouse, and then we have the mouse that runs in circles, which is called the waltzing mouse. Okay, and that's going to be represented with a lowercase r. So gene number 1 is going to be the running type, or running style. Normal running, capital R, and waltzing, a lowercase r. And so that is actually going to be gene number 1. Gene number 2 let's see, it says black hair is dominant over brown hair. So we're going to describe that gene as hair color. Black is dominant over brown. And so the black gene will be represented with a capital B and will be dominant. The brown gene will be represented with a lowercase b and will be recessive. Remember, the reason that we used B, even though they both start with a B, it's really because we needed to name them after the dominant trait. And we needed them to be the same so that we could recognize that they are the same gene. All right, so that is gene number 2. And now it says, cross a heterozygous running homozygous black mouse with a homozygous waltzing homozygous black mouse. Oh my goodness, that is so many words. And so the question is, Mr. Willis, what do I do? How do I do that? We work through one little piece at a time. So we're representing the parent cross, and that's going to be, we're going to write the genotype for one, and we're going to cross it with the genotype for the second one, and we're just going to go through one thing at a time. So it says cross a heterozygous running, okay, so running, not waltzing, but running, which means and it says heterozygous. So heterozygous, of course, means big, little. So there's a big, there's a little. Heterozygous running mouse that is homozygous for black fur. Okay, so black fur and homozygous. So black fur is with a capital B, and it'll be homozygous, meaning that they will be the same. So heterozygous for running style and homozygous dominant for hair color, or homozygous black. All right, let's see what the other one says. With a waltzing homozygous black mouse. Okay, waltzing. Now, since waltzing is a recessive trait, the only way it can have a waltzing phenotype is if it is homozygous recessive. So I'm going to write that that way. And let's see, what color? Homozygous black. So again, capital B, capital B. And so a homozygous waltzing, homozygous black mouse. All right. So that is how we write the gene trait, the alleles, and the parental genotypes. Take a moment and do that for parts B and C. All right, welcome back. We're going to flip to the back now, and we're going to write possible gamete combinations. So we're going to learn how to take our genotypes and actually write gamete combinations from them. So this first one is going to be capital R, and I'm on the back looking at just below the paragraph of information, and I see something that is heterozygous for the R trait and homozygous recessive for the T trait. We don't know what those traits are, we just have that genotype. Okay, and what we're going to do is we're going to use what's called the FOIL method. That stands for first, outside, inside, last. So the first trait, and now when we create gametes, we only pass on one of each gene type, or one of each gene trait. So this organism could pass on, this parent could pass on a capital R or lowercase r. But that capital R could pair with this lowercase t, or it could pair with that lowercase t. And this lowercase r could pair with this t or with that t. And so we have to look at all four possibilities. So let's look at the first one. Capital R, lowercase t. The second one, the outside, is capital R and that lowercase t. The third one is the inside, which will be little r, little t. And the fourth one will be little r, little t, again, the last genes. So little r, little t. Okay, and so those are the possible gametes, and we would write those along the side of a dihybrid Punnett square, which of course would be 16 squares, a 4x4 square. And then we would have another parental genotype that we would get 4 gametes from, that we would write on that side, and then we would fill in our cross. All right, so let's look at one more. So now we're going to look at capital G, capital P, lowercase p. And again, we'll look at first, which will be capital, outside, which will be capital, lowercase, inside, capital, and lowercase, or capital, lowercase. And again, those are our possible gametes that could be passed on, and so we'll write them along the outside of the Punnett square. Okay, and then we would fill that in, which we'll do some more of later. Take a moment and also complete questions C and D of that question. All right, now we're going to move on to the problem-solving steps, where we will solve a complete 2-factor or dihybrid cross by ourselves. I'm actually going to bring in a pre-made 4x4 square, and let's write down some information as we go. So if you ever get lost or get stuck, just follow the steps on the problem-solving steps 1 through 8, and just work through one step at a time. In peas, green seed color is dominant to yellow. Okay, so green seed color, so I'm going to just write up here green, and this is going to be for the trait seed color. Green seed color, capital G, is dominant to yellow seed color, which we will represent with a lowercase g. All right. The other trait that we're going to be examining, it says, and round seeds are dominant to wrinkled seeds. And so that is going to be seed texture. Okay, and round is dominant to wrinkled. And I'll name that after the dominant trait, so I'll put a capital R, and wrinkled will be with a lowercase r, because we have to use the same letter. All right, next we need to, so that is how we would fill in the gene information and the gene trait information. The next part that we need to look at is we need to write the parental genotypes. Let's see. It says, predict the results of a cross between 2 plants heterozygous for both seed color and seed shape. Okay, heterozygous for both. So there's heterozygous for seed color, there's heterozygous for seed shape, and so there is one parent. I'm going to write my other parent over here again, heterozygous for seed color, heterozygous for seed shape, and those are the 2 that I will be crossing. Now, we need to determine the possible alleles, or excuse me, the possible gametes. Remember, we will do first, outside, inside, and last. And so first is going to be capital G, capital R. Outside will be capital G, lowercase r. Inside will be little g little g capital R, and last will be little g little r. Since this is the same genotype, it will have exactly the same possible gametes. And so we will just write those along the top. I made a mistake there. That's a lowercase g, capital R, and little g, little r. So take a moment and fill in your dihybrid squares with the correct. I'll do the first one, G, G, R, R. And so take a moment and fill in the rest of the squares. Okay, welcome back. Hopefully you have got those filled in. Take a moment and check and be sure that it matches with what I have filled in there. Okay, and so now what is left is for us to chart our genotypes and phenotypes and to answer the question. So for example, the first F1 genotype says capital G, capital G, capital R, capital R. This is the only one of those that I see, and so I would put a 1 next to that. Then capital G, and capital R, lowercase r. Well, I happen to see 2 of those, so I'll put a 2 out beside that. All right, and then take a moment and fill in the rest of those genotypes. For phenotypes, we'll either be counting our green round, green wrinkled, yellow round, or yellow wrinkled. Those will be our 4 phenotypes that we'll be charting. And then finally, the question says, what is the chance of getting offspring with yellow wrinkled seeds? Well, when I look, first of all, I need to find yellow. All these are green because of the capital G's. Okay, here are my lowercase g's. So here are my yellow. And so the question is, which ones are yellow and wrinkled? That's going to be round, round, round. Here is a yellow wrinkled offspring. And so that is the only one out of all 16 that could possibly be yellowed and wrinkled. So the answer is that there is a 1 in 16 chance of that happening. All right, that brings us to the end of our notes on 1-factor and 2-factor crosses for part 1. I hope that was helpful, and have a great day.